Hello everyone, welcome to my channel. So in the previous videos we had covered the part 1 and part 2 of diabetes mellitus, that is we had covered the introductory part and the classification of diabetes mellitus. Now we will read about the pathogenesis of the type 1 diabetes mellitus in this video. So coming to the pathogenesis of the type 1 diabetes mellitus, the type 1 diabetes mellitus accounts for 5 to 10%, approximately 5 to 10% of all cases. The most commonly the type 1 diabetes mellitus occurs during the childhood, but it can occur in any age group. So it should not be referred to as juvenile diabetes since it can occur at any age group. So it should not be referred to as juvenile diabetes, but it is most commonly seen in childhood. The type 1 diabetes mellitus is an autoimmune disease. It is an autoimmune disease in which the immune effector cells of our body react against the endogenous beta cell antigens, resulting in islet destruction. So since the immune effector cells of our body here are reacting against the self antigens of our body, therefore it is an autoimmune disease. Now coming to the pathogenesis of type 1 diabetes mellitus, it includes two factors. First, the genetic susceptibility, which includes the genetic factors. The most important locus in the genetic susceptibility in the HLA genes, and there are also certain non-HLA genes. The second factor are the environmental factors, which mainly include the viral infections. So we will read both of this in the subsequent slides. So first, coming to the genetic susceptibility, more than 30 susceptible loci have been known which are responsible for type 1 diabetes mellitus, but out of this the most common locus is the HLA gene cluster. So first we will see about the HLA gene. The HLA gene cluster is located on chromosome 6p21. So it is located on the short arm. The HLA gene cluster is located on the short arm of chromosome 6p21. Now the subtypes of HLA gene which are mainly involved in the susceptibility for type 1 diabetes mellitus are DR3, DR4 and DQ8. These are the important subtypes involved in the susceptibility for type 1 diabetes mellitus. So this is about the HLA gene. Now coming to the non-HLA gene, the polymorphism of the non-HLA gene, will affect the negative selection of the reactive T cells. What would happen? Due to the negative selection, the immune cells which were reactive against the self antigens of the body would get apoptosized. Otherwise, what would happen? If such cells would not have been apoptosized, it would react against the self antigens of the body, which would result in autoimmunity. So via the negative selection, such autoimmunity is normally prevented. But since here the negative selection is affected, therefore such reactive T cells which can act against the self antigens, self beta antigens of our body will still persist. Such reactive T cells will persist, which will result in destruction of the self beta antigens, which would affect the level of expression of insulin. So the polymorphism of the non-HLA gene would affect the negative selection of the insulin reactive immune cells, which would affect the level of expression of the insulin. Polymorphism of CTLA4 and PTPN2, these are the non-HLA genes whose polymorphism are involved in the alteration of T cell selection and T cell regulation. So the non-HLA genes are mostly via their polymorphism altered the T cell selection and the T cell regulation. So this is about the genetic susceptibility, the HLA gene, the most common subtypes being DR3, DR4 and DQ8, and the non-HLA genes. Now coming to the environmental factors, the viral infections with Coxsackie B, mumps, rubella or the cytomegalovirus are mostly involved in the diabetes mellitus. So how do these viruses induce the autoimmunity of our body? There are three mechanisms which can explain the role of the viruses in the induction of autoimmunity. So let's see one by one. The first mechanism is the release of hidden or sequestered antigens. What happens? Normally the beta cell antigens are hidden or sequestered within the islet cells. But once the viruses invade our body, this viral infection causes injury to the islet cells and inflammation of the islet cells, as a result of which the beta cell antigens which were sequestered or hidden within this beta cell is now released. This beta cell antigens which were sequestered initially in the islet cells due to the injury by the viral infection, these are now released. So this beta cell antigens are now released, so the immune system of our body can easily recognize this beta cell antigen, as a result of which the reactive T cells of our body would get induced, resulting in induction of autoimmunity. So the viral infections via injuring the islet cells would release the sequestered antigens, which would result in induction of the autoimmunity. So this is the first mechanism. Now coming to the second mechanism. The second mechanism is the molecular mimicry. What would happen? The viruses will enter the body, and these viruses would cause release of certain proteins. Now these proteins would mimic the beta cells. These proteins would mimic the beta cell antigen. So since these proteins mimic the beta cell antigen, so what will happen? The proteins being foreign particles, so against this our immune system will generate a response. Our immune system will generate a response against these proteins. But since these proteins resemble the beta cell antigens, so this immune response may cross react with the beta cell antigens, resulting in destruction. So the response which is generated against the viral proteins, since these viral proteins resemble the beta cell antigens, therefore such response may cross react with the beta cell antigens, resulting in self tissue destruction. So this is the second mechanism, which is the molecular mimicry. Now coming to the third mechanism. The third mechanism is the sharing of antigenic epitopes. So here what would happen? Suppose a viral infection initially means in the earlier life occurred in the body. So this virus has entered the beta cells and it persists in the beta cells. Now what will happen? There is a reinfection by another virus. But these two viruses share the same antigenic epitopes. Initially the virus which had entered and is still persisting in the beta cell, its epitope is similar to the one which has caused reinfection. So now when the body will generate an immune response against this virus, it can also lead to destruction of the beta cells, since the virus sharing the epitope with the persisting viral antigen is present within the beta cell. So the immune response can destroy the persisting the immune system can destroy the beta cell as well. So the sharing of the antigen epitope, since earlier in life the virus which had caused an infection and presently the virus which is causing an infection, these two viruses share the same epitopes. So the immune response generated against the present virus can also be reactive against the one which had affected earlier and is still persisting in the beta cell, therefore leading to beta cell destruction. So sharing of antigen epitopes is the third mechanism which can result in induction of autoimmunity. So these are the three mechanisms via which the viruses can induce the autoimmunity. So this is all about the environmental factors, that is the viral infections which induce the autoimmunity via the release of hidden or sequestered antigens, molecular mimicry, and sharing of antigenic epitopes. Now coming to the mechanism how the beta cell are being destroyed. So the mechanism by which beta cell is being destroyed is, first of all, there is a failure in the self-tolerance in T cells. So what is tolerance? Tolerance is a state in which an individual is incapable of developing an immune response against the self-antigens. So normally our body do not induce an immune response against our self-antigens. So this state in which the body do not produce an immune response against self-antigens, this is what is referred to as tolerance. So in type 1 diabetes mellitus, what happens is the self-tolerance is T cells is failed. So the T cells are no more tolerant, so it will react against the self-antigens. Now why is this failure of self-tolerance occurring? It may occur due to defective clonal selection of self-reactive T cells in the thymus. We had earlier read about the negative selection. Then due to defects in the functions of regulatory T cells. So since there is defect in the function of the regulatory T cells, therefore the regulatory cells means the affected T cells are resistant against suppression by the regulatory cells. So due to the defect in the function of the regulatory T cells, there is a resistance of the affected T cells due to which it is not suppressed by the regulatory T cells. So due to these reasons, there is a failure of self-tolerance in the T cells due to which it can now act against the self-beta antigens. So now due to the genetic susceptibility and the environmental factor, there will be failure of self-tolerance in the T cells, and due to this failure of self-tolerance in the T cells, since it can now affect the self-antigen, since this self means now this reactive immune cells can react against the beta cell antigens, so it can result in destruction of the beta cells. Then due to the Th1 activation, there will be release of certain cytokines like interferon gamma and TNF. Interferon gamma and TNF. These are the cytokines which are released by the activated Th1, which can lead to beta cell destruction. Then the CD8 plus CTLs can directly cause beta cell destruction. This can directly cause destruction of the beta cells. And lastly, since autoantibodies will also be formed, from the tolerant B cells, the autoantibodies formed will also cause destruction of the beta cells. So due to the failure of self-tolerance in T cells, due to release of cytokines like interferon gamma and TNF from the activated Th1 cells, due to direct stimulation via the CD8 plus CTLs, and due to release of autoantibodies, this all will cause beta cell destruction. So ultimately, due to beta cell destruction, what will happen? There will be absolute deficiency of insulin. Why? Because insulin is released from the beta cells. So since there is a destruction in the beta cell, so obviously there will be a deficiency in the insulin. So the genetic susceptibility and the environmental factor will cause the failure of self-tolerance, Th1 activation, CD8 plus CTLs will directly affect the beta cells, and release of autoantibodies, all of which will cause destruction of the beta cells, due to which there will be a absolute deficiency of insulin. So these are all about the pathogenesis of the type 1 diabetes mellitus. Thank you.